Now, if this is the first time you're seeing me, you're doing it wrong, because this is the beginning of the second part of our lecture course. In the first part, we discussed the basic underlying mechanisms of apoptosis. And in this second part, we will discuss how apoptosis and cancer are related, and how cancer research and apoptosis research interact, and how therapies, therefore, should be focused on these aspects. I hope you will enjoy the second part of our lecture.
Good. Let's talk about therapy-induced apoptosis. Now, one of the hallmarks of cancer, and if you work in oncology or any related field, you will know this slide, you will know this review. It is the standard review we all have read and quoted in our papers, in our thesis, in everything. One of the key features of cancer cells is their increased resistance to cell death, or the increased resistance to apoptosis specifically. Now, from the get-go, we create a conflict, because we try, as we discussed in the last lecture, to induce apoptosis by radiation and chemotherapy to overcome cancer, and yet the very definition of cancer means that these cells are already highly resistant to apoptosis. So we try to kill cells via a mechanism we know they don't respond to particularly well. However, if you just look at the sort of list here I put together of all the chemotherapy or radiation I could think of the top of my head, and then if you add the mechanisms by which those substances or those methods kill the cells, you find that most of them kill via induction of apoptosis. So DNA damage causes apoptosis. So whether you break the apoptosis, whether you break the DNA, whether you cross-link the DNA, or whether you cause DNA adducts, that normally causes apoptosis. What also causes apoptosis, if we inhibit topoisomerase, which unwinds DNA during replication, if we stop the unwinding, that DNA gets broken. So that's apoptosis as well. Radiation does two things. Radiation either directly or indirectly damages DNA. So that's apoptosis as well. Mitotic inhibitors are believed to mainly work via mitotic catastrophe. That's a form of programmed cell death which isn't apoptosis or isn't classical apoptosis. So we could exclude those two. However, the main bulk of treatment options induce apoptosis. And I'm sure you can think of other methods and other ways. Again, we've got a forum on the webpage. We can discuss it there. So the question is, of course, we try to induce apoptosis in a population of cells which are resistant to apoptosis. If we want to overcome that resistance, we first have to understand why it's there in the first place. And basically, the more genomic data we get, the more sequences we have of tumors, of different tumor types, of different tumors in different patients of the same type, we know there's a lot of mutations going on. And there's not really the specific cancer gene. But basically we have two types of mutation. We have mutations where proto-oncogenes get activated to oncogenes, that's dominant, or where tumor suppressor genes get inactivated, and that's normally recessive, where you need to, and we have DNA repair genes which are sometimes added. So, and in this field, it is normally, very big normally, the tumor repressor genes, which are there to induce apoptosis when something goes wrong. So basically, cancers have developed many different methods of not dying by taking away key components of the program which is supposed to kill them. And this list is incredibly complex, and part of your exam will not be to memorize all the numbers, but what you have here is the mutational landscape of different tumors. So you have different tumor types listed over here, and you have different genes listed here, which have been clustered in genes with different functions or in families. And the same here, tumor, genes, functions. And you see that there's a very random distribution. Those numbers indicate the percentage of mutations found, or how many tumors are mutated for that particular gene. And you find a huge variation. Not all of them, but many of these mutations are different methods of how the cancer avoids dying. If you want to therapeutically treat cancer, unless you are a fan of personalized medicine where we sequence every single cell in a cancer, we want to aim for something which basically is mutated in many cancers. And the absolute classic example is highlighted here, which is p53. p53 is also called the guardian of the genome. It is the key protein which links DNA damage response to apoptosis regulation, and it is basically induced by DNA damage or by anything going wrong in the cell cycle or by the absence of oxygen. And then, depending how strong the damage is or how strong the induction is, p53 leads to a stop in proliferation, so it stops the cell cycle, gives the cell time to repair itself, and once the repair has occurred, it sort of restarts the whole process. If the damage is too strong, the repair takes too long, p53 then sort of makes the decision that suicide is the better option. So p53 is kind of gatekeeper. It regulates the response. And there's also a review on the webpage for you to download and learn more about p53. Now, importantly, p53 is mutated in about, depending which study you believe, let's say 50% of all tumors. It is the most frequently mutated or inactivated gene in cancer. Now that would make it an ideal target. We reactivate p53 in the tumors and therefore resensitize the cancer for apoptosis. Now, cancer cells are already really fast dividing. Otherwise, you wouldn't have the tumor in the first place. So you have fast dividing cells, on average. You reactivate the control mechanism which goes, hey, wait, it's all going too fast here. And that should actually kill the cancer cell. It's a very good idea. There's a lot of research going on. There are substances which either aim to block the inhibitor of p53. So basically you take away the brakes and p53 gets activated. And there are substances that chaperone p53 mutants back into the right configuration. So the idea is p53 gets mutated. It's therefore the wrong shape, can't do its job anymore. You add a drug which sort of forms a shape around the p53 and forces it to go back into the right configuration and therefore activates it. And there are even clinical trials where you try to reintroduce wild type, normal p53, into cancer cells via viruses, for example. That's all very good. There's a tiny wee flaw in the plan, and that is it doesn't work. So far, every clinical trial which was focused on p53 was not necessarily a failure, but not such a good result as to be expected by the frequency of p53 mutations found. So, although p53 seems to be target number one, in research it hasn't lived up to its promise. Now, another interesting target, and one I'm slightly biased towards, because I did work a lot on it, is the PI3 kinase AKT-mTOR survival cascade. So if you take that signaling pathway together, either the inactivation of the negative regulator P10 or the activation of the kinase, it is in many tumors fairly often that this pathway is activated. Actually, P10, depending again on which study you read, is after p53 the second most mutated protein in cancer. And some people have even started calling it the new guardian of the genome, because we're really not very creative with names. This is a PI3 kinase signaling cascade. It is, like the slide I showed you about the apoptosis pathways, hugely complex. But you will also find some of our old friends again. So basically what we have is PI3 kinase gets activated. That leads indirectly to phosphorylation and activation of AKT. That activation is blocked by P10. P10 fairly frequently not present in cancer, so AKT gets more and more activated, and AKT regulates many of the key players we have talked about before. So here we have members of the BCL2 family, which get blocked. So these are the pro-apoptotic members. They get inactivated by AKT. We have the XIAP protein, which gets activated by AKT. And we have caspase 9, which gets inactivated. So again, basically what we have here is a repeat of the apoptosis signaling cascade, now blocked by AKT.